Hi, my name is Cerveny. I'm a cell and developmental biologist here at Reed College, where my lab studies how cells transition from proliferation to differentiation in the developing nervous system. I'd like to take this opportunity to show you a quick tour of my lab, to show you the zebrafish that give us the embryos that we work on, and to show you the microscopes that we use for much of our experimenting. So here we are in the main lab. First, I'd like to show you around the zebrafish room. These little tropical fish give us so many embryos each time we pair them that we can do many, many experiments at the genetic scale and at the cell biological scale. We have wild-type fish, over 15 different transgenic lines that allow us to see blood cells or parts of the nervous system that are responsive to different signaling pathways, or even that just outline cells within different parts of the nervous system. We also have a number of mutants in our facility. We have about 10 different mutant lines of zebrafish, some of which we have generated ourselves. And we use all these different zebrafish to generate embryos that we can study. How do we get the embryos? Well, zebrafish reproduce relatively quickly. You put a male and a female together in a tank in the afternoon. By the next morning, they will have laid eggs, and those eggs can be easily collected and then studied over the next several days to watch the nervous system develop in real time. Back in the main lab, you can see that we have dissecting microscopes where we do our embryological manipulations, injections, dissections, transplantations, as well as just looking at embryos after we've done particular procedures to them. But my favorite part of the lab is through this door. It's the microscope room. And in here we have fluorescent dissecting microscope, fluorescent compound scope, and one of the most important pieces of equipment that we use to study zebrafish development in real time is this laser scanning confocal microscope outfitted with a temperature-controlled box around it so that the zebrafish can develop normally and we can film that process. So I hope you enjoyed that short tour of my lab. If you have questions about the research that we do here, are interested in ways to get involved, or have questions about what it's like to be a student at Reed College and engage in novel research alongside your professors, feel free to reach out. You can contact me with the information below. I look forward to hearing from you, and I hope to see you in the fall.